Garrett, Tommy, and Piper's computer-aided drug design PowerPoint. So let's get right into it. Before we talk about computer-aided drug design, we have to talk about computer-aided design. And you cannot talk about that without talking about Dr. Patrick J. Hanratty. Dr. Patrick is considered the father of CADD, and in 1957 he created Pronto, and this is the first commercial numerical control programming system. 6 years later, in 1963, Hanratty partnered with IBM and GM to develop Design Automated by Computer, or DAC. These two inventions led to CAD software for all industries, including computer-aided drug design. CAD is the use of computer-based software to aid in design processes. It can be used to create 2D drawings or 3D models. And computer-aided drug design simulates and analyzes molecules for discovery and advancement of drugs. Computer-aided drug design is a constantly evolving field with many advancements and inventions, but we're going to focus on 5 significant moments between the 1960s and 2000s for this part of the presentation. That is the development of molecular modeling, structure-based drug design, virtual screening, fragment-based drug design, and personalized medicine and drug repurposing. During the late 1960s to 1980s, computational techniques such as molecular mechanics, dynamics, and quantum mechanics are developed, and they were notably developed at MIT. Carol K. Johnson introduces the ORTEP program in 1965. That is the visualization of molecular structures using X-ray. And then David and Jane Richardson's 1970s pioneering work in visualizing protein structures entirely on a computer. These techniques, in addition to Carol Johnson and the Richardsons' inventions, laid the foundation for computer-aided drug design in much the same way that Pronto and DAC paved the way for computer-aided design in general. These advancements allowed scientists to simulate molecular structures and interactions, and this led to insight into drug interactions and accelerates drug discovery. Structure-based drug design, or SBDD, emerged in the 1980s. It is the rational design of drugs based on 3D structures of proteins. Precise shapes and interactions of proteins allowed researchers to identify binding sites and design molecules to interact with them. This provides insights into disease mechanisms at a molecular level, and these 3D structures lead to development of specific and potent therapeutics with reduced side effects. Virtual screening came about in the 1990s, specifically 2 methods of virtual screening, that is ligand-based screening. A ligand is just a small molecule that attaches to a larger molecule. And with ligand-based screening, you can identify drug candidates based on structural similarity of known active compounds. The other method was structure-based screening, and that's when you use 3D structures of proteins to identify fits to the protein's active sites. You can computationally screen large libraries to identify potential drug candidates, and this significantly accelerates drug discovery process. These large libraries can get up to the millions of compounds, and these can be quickly screened, and this lets researchers prioritize compounds for experimental testing based on binding affinity. If you look over to the right, you'll see a little infograph about 2 target proteins from the COVID-19 virus that this group of researchers were trying to find compounds that interacted with them. And you can see that they pull from approved and clinical trial drugs, and it goes through 3 levels of screening, which is shape similarity, docking, and that's when you do simulations to see if the structures will dock correctly, and then you have interaction similarity. And as you can see, they start out with 6,200 compounds roughly and end up with 38 compounds. So instead of going through each one of those individual 6,000 or so compounds, they can run it through the screening process and identify the compounds that will interact with the target proteins in some way. And then from there, they can identify the ones that will react positively to them. Up next is fragment-based drug design, or FBDD. It became an alternative to traditional screening methods in the early 2000s. With this, you screen a small library, just hundreds or thousands of compounds in comparison to the millions of compounds. And more specifically, you screen a small library of low-weight molecule fragments to see if they bind to the target. And this offers a starting point for development of drugs. If you look over to the right, you'll see a jigsaw puzzle with a few buttons on it. Basically what this is representing is that if you look at the blue one inside the jigsaw puzzle piece, say that connects well, you can then build out the molecule from there once you have a starting point. This allows for more efficient exploration of chemical space. It really helps with the designing of molecules, and it improves the potency and selectivity. And this leads to the discovery of novel drug candidates and therapeutics. And lastly, we have personalized medicine and drug repurposing. These are pretty self-explanatory, but it started in the early 2000s, and it's picked up lots of momentum in recent years due to advances in CADD. With personalized medicine, you take individual patients, you analyze them, and you tailor the treatments to that patient. And some great benefits of this will potentially lower healthcare costs and improve patient outcomes. And with repurposing, you identify existing drugs that would be effective for different diseases. And this turns reactive healthcare into proactive healthcare. Now I'm going to pass it along to Tommy to talk about current uses.
Now we are on to my section, which is the examples of the current uses. Here we are going to start with ion channels, which are a very important part of the human body because what they do is they control the flow of the electrical signals throughout the human body. So any signal that will, the signals, they are responsible for muscle contraction, immune responses, hormone secretion, really anything like that is anything the human body does is controlled by an electrical signal, which flows through the ion channels, as showed here on the right. This right here, the blue part, is the ion channel. This is the cell membrane, and this is kind of the electrons, or the signals, flow through here inside the cell, and they tell them what to do. And so there is a very promising future in drugs that target ion channels because there are very many issues that are caused by defects in ion channels or disruptions in the flow of the ion channels, such as neurological diseases, cardiovascular diseases, chronic pain, muscular degradation such as ALS. Really anything like that it can be possibly treated in the future by drugs relating to ion channels. And there are over 300 different ion channels in the human body, but there are very few drugs that specifically target the ion channels. And so if we were able to get more drugs to treat more of them and more issues with them, that will open so many more doors for treatment of diseases we find to be currently incurable because they are caused by a part of the body we cannot treat. And how CADD will help with this is it will use the structural structural drug design part, where it will 3D model the proteins and the molecules of the drugs and see how they bind with one another, and that will vastly increase the speed of the drug discovery, especially in the early stages, because you need to see what can bind to what before you do any sort of testing. Especially so in recent years, we have discovered much more actual structures of the ion channels, which will make it far easier to create more drugs that will actually affect them and help to mend or repair any sort of dysfunctional ion channels in the human body. And that opens so many doors for development. And then the next most relevant subject we could find was anti-venom development, because out of the 745 venomous snakes in the world, not even half of them actually have an anti-venom developed for their venom, for their toxins. And that is because snake venom contains hundreds of different proteins that are toxic to humans or just animals, which they can cause all sorts of, all those proteins can cause all sorts of, like, malaise, so to say, in the human body, disturbances such as inflammation, pain. It'll make you continuously bleed. There's gonna be neurotoxins. Really, all sorts of toxins are caused by the individual proteins, and when you multiply that by hundreds, you see where it starts to get tricky, because in the drugs, in the antivenoms, you have to have, say, compounds that neutralize each and every one of those proteins so that the symptoms can be stopped or neutralized. And so in recent years, there have been developments in specifically bioinformatics tools within CADD, which is running simulations and algorithms and all that, to kind of mine through the snake venoms to, like, single out all the proteins, kind of sequence them, see what's in them. Because if you see this right here on the right of the screen, this is this is a visualization of what snake venom looks like. Each and every different color of strand here is a different protein within. And so, with how twisted together it is, all the jumbles, you can see how difficult it is to, like, to single out each and every protein with with only humans doing it. But with all the recent advancements we have made in computer aided drug design, it opens the door for so many possibilities to characterize all the venoms and toxins within a snake's venom. And we can use, like, computational techniques to predict what the toxin will attack, and thus, like, find how to bind, like, compounds to the venom to neutralize it and to make it so where it won't impact you. And so that is it for my section.
In this section of our presentation, we are going to be looking at what the future holds for computer-aided drug design. In its current state, the industry is headed towards quantum computing for that design, and that's due to all of the benefits that come along with quantum computing. But before we get into that, we're going to go and take a brief history of quantum computing. So, the first quantum computing algorithm was successfully demonstrated in 1994 by a team led by Isaac Chuang at the Los Alamos National Laboratory. This demonstration was performed using a 2-qubit quantum processor. For an idea, this processor, the newest processors out there, have about 133 qubits. In 2013, Google announced their first quantum computer using the most modern tech available at the time. And fast forward to 2019, Google claimed quantum computing supremacy and continued to push the tech forward. That being said, Google is absolutely not the only one that is using quantum computing. While Google has one, IBM also has one, and a few other of the major tech companies, have them as well. With that brief history out of the way, the reason we are moving on to quantum computing for computer-aided drug design is because it's a lot faster than modern computing. You can run far more complex simulations, you can run more simulations at once, and all of these are run at much faster speeds than current computing methods. This is all done by the fact that they use a qubit system versus a bit system, which is currently being used by modern computers and supercomputers. The way the bit system works is it's binary. It uses a 0 or a 1 to indicate data, where the qubit can indicate 0, 1, or both at the same time. These are done using superimposed electrons that are they are measured. You By measuring one of them, you can get the measurement of the other electron that it is partnered to. It is always opposite to what it is. And this allows quantum computing to effectively be both process both as a 0 and a 1 at the same time, which exponentially increases the speed at which you can process. And that is our presentation on computer-aided drug design, the history, the current, and the future.